All right, so let's talk about the nervous system as a whole. First thing that you want to focus on is the fact that it's divided into two sort of systems. There's what we call the central nervous system and the peripheral nervous system. Diagram here shows it very nicely. Central nervous system includes your brain and your spinal cord. These are the two major organs of the nervous system. So it isn't like you got a dozen organs like the digestive system or something. You've got two basic organs here. From that standpoint, it doesn't look very complex, but the complexity in the nervous system is not at the gross structural level. It's down at the cellular level where most people can't see. Ancient people were enamored by all these various organs in your human body, thought they were very special. Nervous system was very often just totally disregarded. My family and I went to an Egyptian exhibit a number of years ago where they showed how they did the, mummifying the body and all, and, organs in here were recognized as very important parts of who you were. And when they mummified a body, they would take all the organs out of the thoracic cavity, and they would actually put the heart back in to the body when it was mummified, but all the other organs in here had special jars to put them in that would sit in little niches in these burial tombs and stuff, and your liver would go in its own special place, and your stomach would go in its special place, and the intestines had a special jar, the lungs had a special, clay jar for them to go in, and you had all these things where they would just scoop out the brain and throw it away. It just looked like fatty, filler stuff in your body. And it would rot in there and stuff. So they would just throw that stuff away. So from an external standpoint, it doesn't look like a whole lot. It's when you get down into the cellular complexity of the nervous system that you really see marvels upon marvels. So, central nervous system, brain and spinal cord. If you're in any kind of medical work, you'll hear people talk about CNS this, CNS that. And they're talking about the central nervous system, as distinct from the peripheral nervous system, which are all the nerves in your body. And there are two sets of nerves. There are nerves that come out of the brain and nerves that come out of the spinal cord. Cranial nerves and spinal nerves. So this is a very simple layout for four basic structural components of the nervous system. The nerves that we're talking about here are really just the wiring of the nervous system. How the central nervous system that does all the action connects itself to the elements of the body that it's in touch with. Okay, so just reiterate, central nervous system is brain and spinal cord, right? Peripheral nervous system, right, are the nerves that connect it out. And in the brain, these little yellow pieces that you see in here clustered around the center there, those are the nerves that are being talked about here. Now, I'm going to divert from your handout for just a moment, just a little bit. I've sort of redone this handout in a number. I'm trying to find just the right way to kind of proceed through this. And I realized, why don't I do this? I've been doing it with all the other systems. I start with a cellular level and then work up to the tissue level and then work up to the organ level. And that's really what we should do here. So you're not going to see notes in this yellow handout for what I'm going to do right here. You're going to have to take some of your own notes. But I think this is going to work because I always do go through the cellular level, but I used to do it kind of later in sort of a context. I think it's just as good to start from the beginning. Okay, so let's do that. The actors of the nervous system. Okay? The living things that make it happen are these neurons. Now, do you remember that one of our 8 cells at the beginning of this class was a neuron? Remember that? Remember, what we were looking at was just this, right? Your handout looked about like this, didn't it? So we were just looking at a small part of the entire neuron. We were looking at the main body of the neuron. Okay? But there are all these little tentacles that were running off in various directions, and they're an important part of the neuron as well. So let's sort of identify various parts of the neuron and see what kind of a role they play here. Sorry about that. Went backwards. Okay, so first of all, the cell body, okay, is this central part that we had in our handout, where you can see the nucleus and the Golgi and the endoplasmic reticulum and the ribosomes and all of the productive factory part of the cell is right here, what we would call the cell body. But the various processes, the various things that are extending from it are very important too. This happens to be the processing center, or at least the processing center of nerve impulses is here. And then there are dendrites, which are these little extensions clustered in and around the cell body. And these are receiving fibers. These are receiving processes. These little pieces are collecting and receiving impulses and messages from other neurons. Somewhere in the entire neuron there's going to be a sending fiber, and here it's shown in yellow, called the axon. And there's typically just one of these, although it may branch further along into more than one piece. But coming out of the cell body, we typically see just a single axon. And this is how this cell body, this neuron, sends its message to the next location. The next location might be a muscle. It's stimulating the contraction of a muscle. The next might be another neuron that it connects to. Maybe it needs to send its signal to the next one. I think it's sort of interesting that the neuron really mirrors the action of the entire nervous system. You know, when you think of what is your nervous system doing, it's receiving data, isn't it? Right this moment, I'm hearing sounds in the room. I'm seeing sights in the room. I'm tasting this little lozenge that I'm sucking on so that I don't have to cough all during this lecture. Okay? I'm receiving data, my skin. I can feel the clothes on the surface of my skin. I can feel the temperature of the room. I am receiving data through the sensory receptors throughout my body, and those are being fed into my central nervous system. It is processing that data. And I am sending out information from me through my voice, through the gesturing of my arms, through walking around the room. I am acting out. I'm giving output. And that's sort of in a very simplistic way what's going on with my nervous system. It's receiving, processing, and outputting data. And that's really, in essence, what this neuron is doing. It is receiving impulses, it's processing those, and then it's sending a signal out. Or maybe it doesn't. Maybe it receives data and it decides I shouldn't send an impulse. It's really very much like much of our digital lifestyle these days. I don't know if you know really what it means to go from analog to digital kinds of things. Really all the digital concept means is that we code all information in a binary code. Now, electronic things know if they're on or if they're off. And if I can code everything in a series of on-off messages, then I can record whole movies in just little tiny bits of data that are either a 1 or a 0. That's basically the entire digital world. Everything that's recorded now is recorded in ones and zeros and combinations of those. And it's a tremendous amount of data, but it's there. And the neuron is very much like that. It's sending messages or it's receiving all sorts of data and then sending others on. So the neuron is sort of in miniature, it mirrors the entire nervous system. It mirrors sort of your computer system at home, right? If you think the way of how your computer system works, it's modeled on the way our nervous systems work, right? The cell body would be the dendrites here would be the input. If I have a computer system, it's not going to do anything unless it starts receiving some kind of data. So I might type on a keyboard, or if I'm playing a game, I might move a joystick. If I'm drawing something, I might have a tablet and I might draw some things, or I might have a storage device, and some of the input is data that comes out of a storage device. But it goes into a computing portion, a processing area. And it's processed. If I move the joystick, that signal is received, and there's some sort of a program in the computer that then translates that and says, oh, if he moves the joystick this way, then I have to adjust the picture on the screen this way. If he pushes it that way, I've got to make the little character run over here. Right? And so there's going to be output as well. So we've got informational systems here and in our computers, and they all sort of mirror the same sort of idea where there's input, processing, and output of information. So this is sort of a typical neuron. Now don't think this is the only neuron there is. Okay? There are okay. So the basic neuron that we were just looking at is what is known as a multipolar neuron, because it can receive information from many different poles or many different dendrites. You can have many, many, many other neurons connected into this one, giving it information. See, neurons make decisions just like wise people do. You know, if you're a wise person and you got a heavy decision to make, what do? You flip a coin? What do? You get information, right? You talk to many people. You get good counsel. Okay? Good decisions come from good information. You know, a lot of the poor decisions that people make is because they're talking to the wrong people. Oh yeah, they might be their friends, but they may not know what they're talking about. They've got some wild ideas or prejudices or whatever, and so they come up with very, very poor counsel for people. So neurons typically take input from many, many different sources in order to make their good decisions. So this is a multipolar. Here's a unipolar, or what I should say is bipolar. It's got one input, one output. This is what we call a bipolar neuron. It's much more simplistic. Here's a neuron that happens to be over in the sensory camp where we really don't have dendrites as such. This is the kind of a neuron that's really just sending its signal straight through. Doesn't need to process. The signal goes right by the processing center. And we think of this as sensory because I really don't want to try and interpret my senses out here in my body. I want the information coming from my senses to just go straight through into my brain. My brain will do the interpretation, thank you. So these kinds of neurons don't run into a processing center and then a decision is made. These are just kind of wires. Okay? These are unipolar, just running a sensory signal straight through into the central nervous system. So there are a variety of types of neurons. You don't want to get the picture that every neuron looks necessarily like this. But we do have the same principle. We're sending some sort of signal. We're receiving, there's input, and there's output. And there may be processing in between, or there may not. But somewhere processing is getting done. This may send a signal straight through because the processing is going to be done in the central nervous system. These type of neurons generally fall within the central nervous system. These type of neurons generally fall outside in the peripheral nervous system. Now, I think it's so interesting then to think all that I am, all that I think, all that I feel, my decision making, my preferences, my appreciation for things. Ever stop and look at a sunset? I think 2 nights ago, I was kind of driving, I dropped one of my kids off somewhere. I was driving home and I just kind of came over the hill. Sun had just gone below the hills over here. There were like 3 or 4 clouds up in the sky, and they just had these golden edges on them that were just brilliant. You know, I just had to stop for a minute and just look. What is that? You know, what was going on in here? It was neurons firing. You know, and the neurons, there are billions of these neurons woven into networks. You know, sort of things kind of like this, where either lots of input from lots of neurons connect into some sort of an output, or maybe some sort of simple input is divided up into many outputs. But somehow the appreciation for that sunset is here. Do you know where you parked your car? How do you know that? How do you know that? Where is it in here that gets recorded? You know, how do these little neurons here, you know, how are they able to put that into some sort of semblance so when you leave here today you can go find it? Do you know where you parked your car 2 weeks ago? Probably not. Do you know what you had for lunch, you know, last Thursday? Probably not. Okay? Your brain has ways to remember things short term and ways to remember things long term. Right? Do you know your phone number? Right? Remember your name? Do you know your mom's name and your dad's name? You know, I mean, we've got some long-term stuff and some short-term stuff. All of it is in these clusters of neurons. It's just incredible. Is it interesting to think about your brain thinking about itself right now? Right? You're thinking about how your own thinking is working right now. You know, all that we do in here as students is part of this, isn't it? Right? The whole concept here is, how do I move things from short-term to long-term memory? You'll hear me say this once, or maybe you read something once. How does it get moved into long-term memory so when a test comes or somebody asks you about something, or you've got to remember something so that you can do the next thing correctly, how do you get it in there? How does your brain know what's important? How do you tell your brain, this is important, let's hang on to it for a while? Repetition is huge, isn't it? Repetition is huge. If you don't repeat something, then your brain thinks, this must not be that important. And there are many things today that won't be important tomorrow. What you had for lunch will probably not have an impact on what you do tomorrow. But, you know, do you rely on this as a student? Do you just look at something once, you listen to me, and then you go off and you live your life for a week? You never look at it again. You never repeat it again. You don't tell your brain this is important. Then you try and study the night before the test, and you expect to get or hope to get a good grade the next day. You've been telling your brain all week, this is not important. And then somehow you want to say, oh, the night before the test, oh, this is so important. Help me remember. It just doesn't work that way. All right? You got to let your brain know what's important by whether you repeat it. That's why my number one study tip is always repeat what you've done sometime before you go to sleep. If you hear a lecture, you go through it that same day, you'll remember 2 or 3 times as much as if you try and come back to it another day or 2 away. And just that process of repetition. It's how your brain knows. Every night you have a little janitor that comes through and he sweeps out all the stuff that's useless from the day. Where you parked your car, what you had for lunch, which shirt you chose to wear today. All that stuff that just, isn't going to have any impact on tomorrow. That all gets swept away. And most of this lecture will get swept away. Okay? If you don't do something to tell your brain that this is important. So, and these are simple pathways. Sometimes you have neurons that actually then feed back. Okay, so you can have neurons that feed back on. So there are just incredibly complex organizations of neurons in your brain that result in your feelings, your thinking. Thinking is a part of this. Feeling is a part of this. Right? My thinking. Okay, how I feel about things. Right? And all that I do. I process things, I sense things in my world, and I'm acting out things. Right? All of these things are part of just neurons woven together over and over and over again. And these are pictures or artistic pictures of neurons that are interconnecting in this nervous system. Right? This is nerve tissue. Nerve tissue is one thing. If you remember from our tissue section, we didn't have different kinds of nerve tissue. We just have nerve tissue. And it's a weave of neurons. Now, along with those neurons, there are support cells. When we look down into tissue, we don't just see neurons. Neurons are so targeted and so focused on what they're doing that they can't do everything. They need helper cells within the tissue to help them do what they do. So nerve tissue is this sort of weave of neurons and helper cells. Helper cells are called neuroglial cells, or in many textbooks they're just abbreviated as glial cells. You'll see that too. And either one is really okay to use. Use the term neuroglial or just glial cells. In the central nervous system, there are 4 different types. There are cells called astrocytes. These are kind of what they look like. They hang on to neurons, they hang on to blood vessels. They really form a scaffolding. These are really the ones that form the structure that hold things in place. Neurons themselves are pretty flimsy sorts of cells. So to get them all sort of anchored and organized and set physically in place, we have these astrocytes. Now, along with that, they also form a cellular barrier over the capillaries. I hope you remember from our discussion of blood vessels that capillaries in the central nervous system are continuous capillaries. They form complete walls. And maybe you remember our discussion of the blood-brain barrier, that there's a chemical barrier between your world, your body, and your central nervous system. And it's partly the continuous capillaries and partly a layer of astrocyte-type processes that cover these, so that materials in the plasma have to be processed through 2 layers of cells before they get into the fluid that bathes the central nervous system. So these astrocytes are key players. If you wanted to summarize, you'd say they're involved in support and nutrition, the formation of the fluid that is bathing all of these neurons. And it has to be a very, very specially formulated fluid. And we'll talk about it a little more in just a bit. A second type of helper cell that we constantly see in the central nervous system are called oligodendrocytes. Oligodendrocytes are basically what we think of as insulator cells. One of the interesting things with neurons is typically their axons, their sending fibers, are very long. And so you may have a cell body over here doing something, and it may send a signal to something far, far away from it. And the axons can be very long. The axons of the neurons basically function as the wiring of the nervous system. You can see the nervous system as a series of processing centers with wires running in between. But just like the wires in this building are typically not just bare wires, all basic wiring that's done in buildings is done with coated wires, wires that are covered in a plastic insulation. So majority of the axons within the central nervous system are covered with insulation. And you can see here's a single cell that's wrapping parts of itself around various axons, providing an insulation so that we don't get short-circuiting in the complex weave of cells and axons. There are also what are called microglia. Microglia are defense cells. These are like white blood cells in your body, only we have special cells in the central nervous system that do what white blood cells do throughout the rest of the body. These microglia wander, they're amoeboid-like, they can wander around, they can gobble up dead and dying materials, they can fight infection. These are the defense cells. You can see the central nervous system is really a world unto itself in your human body. It's a separate place with a separate environment, separate sorts of cells. It's a whole unique thing compared to the rest of your body. And then we find cells that line some of the inner passageways of the central nervous system. They're called ependymal cells. They have little cilia on them, and they help to move fluid throughout the system. We have this specialized fluid that we've mentioned that comes from the plasma, but ultimately circulates in and around and through the central nervous system to provide a special environment. And so these cells here are involved in that fluid production and movement. So there are 4 types of helper cells that you would find in the central nervous system. The peripheral nervous system has just 2 types of cells. What are called satellite cells. And if this was a neuron, remember these sensory neurons are ones that we find out in the peripheral nervous system. Well, the cell body isn't in the central nervous system. It's out here by itself, so it has its own protective cells. And so satellite cells do this. right? They cover and protect the cell bodies that are out there outside the central nervous system, right? They look a little bit like this. You can see all these little gray colored cells surrounding the cell body of this central or of this peripheral nerve. So satellite cells kind of wall off and provide a separate sort of environment for these cell bodies that are outside the central nervous system. And then we also have insulator cells out here called Schwann cells. You may hear about these because they're implicated in any number of muscular and neurological diseases that come back to the functioning of the cells. In this cell, the cell body here is probably in the central nervous system, but the axon is running out through the body to typically go to, say, a muscle, causing muscle action. Well, the Schwann cells are like this. They sort of like coat the axon. They cover over the axon and provide an insulation so that I can pack all of these axons together into a nerve, but each one will be insulated from effects of the others. Pardon me. So here in this picture, here's the yellow axon, and you can see two cells. And if you look closely, basically the cell is grown in such a way that it's wrapped its cell membrane around and around and around the axon to provide many layers of insulation to that. So satellite cells and Schwann cells are the two types of glial cells that we'd find out in the peripheral nervous system. So when it comes right down to it, these are the cells, and nerve tissue is just an incredible blend of all these neurons and glial cells. If you don't know what you're looking for, it can look incredibly chaotic. But when you get right down to it, you can recognize, okay, here are these neurons that are connecting to one another, sending signals to one another, and here are all these glial cells providing the extra jobs for them. I always think of the nervous system like the governing structures of a country. The neurons are like the government officials, like your congressmen and your senators, people that make decisions as to the direction the country's going to go. But all of those congressmen and senators, those people all have lots and lots of aides, lots and lots of helpers, lots and lots of people that go get them something to eat or take their laundry out or clean the office or whatever it is. And that's kind of this relationship here. The neurons are the congressmen and the senators in your human body. And there are all these glial cells that are helping to support them. Neurons are making the decisions for this. Now, we've mentioned it over and over again. This nerve tissue cannot exist in your general human body. It lives in a very, very special environment. Environment provided by that barrier, that chemical barrier to the human body. This specialized environment this chemical barrier that we're talking about is this blood-brain barrier. It's provided by continuous capillaries and astrocytes. You saw this in that other picture. What you need to know as well is that there's a physical barrier. Okay, yes, we don't want just anything in the plasma to get into the central nervous system. We have a physical barrier as well. And it's in the form of a set of membranes called the meninges. Okay? Now within these barriers, there's a special fluid that bathes all of the central nervous system. It's known as cerebrospinal fluid. It is what is produced here through these capillaries and astrocytes. The formation of this specialized environment is by way of this fluid that is provided and formed here. This is that chemical barrier, that blood-brain barrier. Now it's enclosed, as I mentioned just a moment ago, this is enclosed or held within the system through a series of membranes called the meninges. And there's a cutaway picture here that shows this set of membranes as they cover the brain. There's a picture where it shows how they cover the spinal cord. There's three membranes for us to learn. They're the Dura Mater, the Arachnoid Mater, and the Pia Mater. Notice that each one of these names has the word mater in it. Any Latin scholars here know what the word mater means? What is it? Mother. Okay? The word mater here refers to mother. Why do I call these three membranes mothers? Okay? What do mothers do for their children? They take care of them, don't they? Right? And these layers are called mothers because they're caring for the central nervous system in a sense. They're protecting it. Mothers protect their children. They nourish the central nervous system. Mothers nourish their children. So the term mater is used in all three of these layers as a reference to the mothering or the caring, the protecting that takes place here. Now this term meninges, this is the collective term for these three membranes. You've probably heard this in reference to a disease, haven't you? Spinal meningitis, cerebral meningitis, or any number of meningitises. But what is the problem in meningitis? Inflammation, infection of the meninges, the membranes, right? It's not, we tend to think, oh, it must be a problem with the brain or the spinal cord, the nerve tissue. No. If mom is under attack, how are the kids going to fare? Right? Not so well. If you attack the membranes, if their care and protection of the nervous system isn't so good, then of course the nerve tissues are affected. And the infection can spread to them as well. But a meningitis is a problem in the meninges themselves. Let's look at these three one by one. Let's start with the Dura Mater. Dura Mater is kind of what it says. It's the outermost layer. Does the word dura sound like another word in English? Durable. If something is durable, what's it like? Sturdy, strong, tough, right? What do you think this is made out of? You ought to be able to guess. We've had enough in human anatomy by now. Dense fibrous connective tissue, right? Dense, irregular, collagen fiber. This is the same stuff you build the rectus sheath out of and the tendons in your body and the ligaments between bones, the tough, flexible stuff in the human body. You've got a very, very strong layer of that enclosing the entire central nervous system. Translate that Dura Mater from Latin into English. What do you get? Tough mother. Exactly. This is tough mother. Right? This is the side of mom that protects her kids. Right? We're seeing the layer around the spinal cord. This is going to be just inside the bones. But think like in the brain. This is coating the inside of the skull, isn't it? I can fracture my skull, but if the dura doesn't tear, my central nervous system is not compromised. I think of it, you ever have a hard-boiled egg? You can crack the shell, but it all hangs together because there's that little membrane on the inside of the shell. Right? It's a lot easier to peel the shell if you can, if that didn't get glued to the egg. But there's that little membrane just inside the shell of a hard-boiled egg, very much like the dura inside the skull. So this is the very, very tough one of the three membranes. Let's go to the deepest membrane. Let's go to the intimate layer. This is the pia mater, and you see it here. Artist did a very nice job, just kind of drawing it very filmy. I think of this like the intimate apparel. This is the lingerie of the nervous system. Like a leotard, this covers every possible surface of the nerve tissue itself. It's the intimate apparel. If there's a groove, like you see here, see how in this spinal cord here, there's a groove running down the front, isn't there? This is folded right down into that groove. The dura doesn't do that. The dura is just this outer basic covering. But the pia mater is right over the surface so that the actual neurons, glial cells themselves don't touch any of the other layers. This is the underwear of the central nervous system. So I've got the lingerie in here. I've got this tough outer covering out here. Between the two is a third layer called the Arachnoid Mater. And I didn't do a very good job. I was going to come back to that, I guess. If you look at this picture, I threw this in here. The Arachnoid Mater in the picture is this in-between one. But if you look here, this is the brain substance here. There's the pia right there. This tough stuff right here is the dura. And between the two, you can see there's a web-like layer, isn't there? Right? And that's where you get the arachnoid from, right? Think of arachnids and you think of spiders, right? Well, you don't have little spiders crawling through your brain, but it looks like they used to be there because there's this web-like layer that leaves a gap. There's a gap here. And there is part of the pia, this black line right here is part of the pia that's connected to the dura, but it's really the web-like parts that are the parts that are significant here. Notice too, you can see there are blood vessels that wander through this space. This web-like area is a space. It's known as the subarachnoid space. Right? And this is where the cerebrospinal fluid is. Right? It's in this little web-like area there. And there are blood vessels wandering through that. And the little webs sort of hold the blood vessels all in place. They're like woven through the web-like stuff. The web-like pieces kind of give a little cushion between the membranes and the surface of the brain too. So the brain and the spinal cord are sitting inside these membranes, sort of separate from everything, but cushioned by the fluid and by these membranes. Takes a lot of pressure, a lot of slamming your head against something to really create problems in here, because everything is covered here in these membranes and it's got this fluid. So, 3 membranes, 3 layers, Dura Mater, Arachnoid Mater, and Pia Mater, that are part of what we call the meninges. There's another term related directly to this that you're going to hear in medical situations, and that's the term epidural. Ever heard the term epidural? Where would you hear the term epidural? Yeah, the maternity ward, right? Labor, all of that. The term, now that you know that there is a Dura Mater, you're probably going, oh, this must have some relationship to the dura. And it does. This picture sort of illustrates it for us. Let's kind of get our bearings here. You recognize this is the body of the vertebra, right? If you're looking at the rest of this spinous process, right? Transverse process. Here's the vertebral foramen, right? In the vertebral foramen, you can see the spinal cord here. There's spinal nerves coming out. So these would be the intervertebral foramina, right? Okay, looking at this closely, you'd see these would then be the meninges right here. So all of this is within this bone, and you can see that this is smaller than the actual cavity, isn't it? The spinal cord and the membranes, the meninges, are smaller than the entire vertebral foramen. And so the body packs adipose tissue, right, in around this so that it all fits very snugly. Well, that fatty area in here, that space between the bone and the meninges, is what we call the epidural space. And the term epidural is used with anesthesia to indicate that the anesthetic is placed in the epidural space rather than into the actual spinal cavity. Before we had all the chemistry to make anesthetics that could do things like this, there was really, if you wanted to anesthetize a large part of the human body, like if you wanted to anesthetize somebody from the waist down, typically what you did was you put the anesthetic into the cerebral spinal fluid. You actually pierced through the Dura Mater and put it in. These days we try not to do that very much. And, years ago they didn't try and do it all that much, but it was one of the few ways that you could do something like that. There's always a danger if you put it in that you might do something to the central nervous system. Anytime you make a hole through the dura, there's the potential for infection to crawl in. So these days, we have the kinds of anesthetics that can seep through the Dura Mater, that can make their way through and cause the anesthesia, and it only has to be deposited in the epidural space. So, I know this was my wife, when we had our first child, she elected to do this, to have an epidural. It was fascinating because then I got to watch. And it's basically what you see. The anesthesiologist works down here in the lumbar area. It's a safer place to work. You should know why by now. You remember how far down your back the spinal cord goes? Does it run down to your sacrum? Where does the spinal cord end in an adult? It was so long ago. Round L1, right? As soon as you finish with ribs, that's about where the tip of the spinal cord is, the conus medullaris. Right there about L1. Right? So down in this lumbar area, you still have nerve fibers running down to their respective exits. But remember, we call all those nerves that are running down through that area the cauda equina. Remember that? And so they choose to put it in here, and it kind of looks like this. The needle doesn't really have the whole hypodermic thing on it. It just kind of looks like this. And I thought the fascinating thing, I could kind of figure out what the anesthesiologist was doing. If you put it between two lumbar vertebrae, you're just going through muscle. Now you go through some skin, so you're going to have a little bit of a push where you go through the dermis of the skin, which is connective tissue. It's tough. When you go through muscles, remember, are just little fibers, and a push between the fibers is not that big a deal. You could see him push the needle in, and then he kind of would slow down, and then you could see him really feeling for the dura. Because the next tough thing you're going to run into is the dura, isn't it? So you could almost sense that he was bumping up against the dura there. And that's where you stop. You're not going to poke it through the dura. And then the thing is just hollow here. You feed a little yellow tube through. Then he could pull the needle out and just leave the tube in there. And then they put a bag of anesthetic onto the tube, and it just dripped the anesthetic right here into this epidural space right there, which could soak in and then perform the anesthesia that you needed. So when people talk about an epidural, they're really making reference to a space outside of the Dura Mater. Okay? Questions? Anything there? Good? Okay. Pardon me. Okay. Let's do this. You should note when you're in the cranial area that the Dura Mater appears as if it has two layers. And there are two layers of connective tissue here. But one of them is actually the layer from the bone itself. I hope you remember from our discussion of bones that every bone is covered with what is called a periosteum, dense fibrous connective tissue that covers the surface of the bone. Well, the flat bones in the skull here are going to have a periosteum over the outside and a layer of periosteum over the inside. So when you combine the periosteal layer pardon me, and the actual meningeal layer, you wind up with an extra thick, extra heavy layer of connective tissue within the skull. So the deeper one is the actual meningeal layer of the dura. So you'll see it's 2 layers thick. Now, the other thing that I want to point out here, notice there's some little triangular spaces between the 2 layers of the dura. Do you see that? Look right up here again. This little space right here is called a dural sinus. And you see one down here in front too, don't you? And it's formed because the meningeal layer is going to fold down between the 2 hemispheres here. And in that folding down between a couple of things, it's just going to create that little triangular space there. And these little spaces are a very, very important part of the circulation of cerebrospinal fluid through the central nervous system. So I need you to make note of these 2 layers here. And you'll see them come into play. I'm going to walk you through the cerebrospinal fluid, and then about the last thing we do is we'll come back to this and see how this fits into the whole thing here. So we should have this concept now that the meninges cover and isolate the central nervous system from the rest of the human body. That they enclose and keep a special fluid within them that bathes and covers the central nervous system. Now, where does this fluid come from? Well, the brain has hollow cavities inside itself called ventricles. The brain contains ventricles. The picture here shows those ventricles in blue. There's 4 of them that you should note in the brain. Just a quick layout of the brain. When you look at the brain, you see this giant cap-like structure to the brain. This is called the cerebrum, right? The big contoured cap-like part. There's a smaller piece posterior and inferior to that called the cerebellum. Cerebellum is here. And you'll note that there's a brain stem or sort of a stalk. If you think of this like a mushroom, this is the stalk right here. And of course the brain stem is what connects to the spinal cord, so that it's all one sort of a unit. Certainly by now, most of you have taken a psychology class. Do you know that people are right-brained and left-brained? Right? And what we find is when we look at this structurally, the cerebrum up here, the largest part of the brain itself, is really deeply divided into 2 hemispheres. Right? So when I do this, you can see all of this is contoured like the outer surface is. This is surface right here. The only place the 2 hemispheres are connected is this white sort of ear-shaped piece of tissue that you see right here is like a telephone cable between the 2 sides. So the reality is that this is more like I have 2 separate brains in my body than I have one. This may explain why I talk to myself sometimes. Right? You ever play some sports activity and talk to yourself? Oh, you stupid, why did you do that? Things like that. So, the 2 lateral ventricles are in the cerebrum. So, you can sort of see here, if you look closely here, you can see there's a hollow space up inside this. If you notice, each part of the cerebrum is sort of shaped like a mitten. And so, this is up in this part, and this little piece coming down this way is down in the thumb area of the brain. So there's 2 lateral ventricles, one in the left and one in the right. In between and down sort of below that, right in the center part of the brain here, is the 3rd ventricle. I always think that looks like Woody Woodpecker, if anybody remembers who that is. You don't see him much these days. But it sort of looks like a bird head. It's kind of hard to visualize these unless you can look at them from a couple of other perspectives. This is a sagittal sort of picture. Look at these 2 pictures. Okay? Here would be a transverse section through the brain. You can see the 2 lateral ventricles, right? How there's one in each hemisphere. And if you look at this frontal section here, here's the 2 lateral ventricles here, and here's the 3rd ventricle down below it, but more central. See how narrow the 3rd ventricle is? Very, very narrow. You're going to see more and more of this today in our lab activity. We're going to actually take these models and take them apart and be able to identify a number of things here. But so I have these 2 lateral ventricles, this 3rd ventricle. There's a little passageway here between the 2. And then after you leave the 3rd ventricle, there's a canal, there's a tube that runs inferiorly from there down to the 4th ventricle, which is right between the brain stem and the cerebellum. There's a triangular space here known as the 4th ventricle. Now, here's the concept for the fluid. Cerebrospinal fluid is produced in all 4 of these ventricles. As it's produced, as it accumulates, the pressure builds up, and it forces itself out of the ventricles and into that subarachnoid space all over the surfaces of the brain and spinal cord. So if you can imagine for a minute that I'm producing cerebrospinal fluid in this lateral ventricle and the other one, as it builds up in there, it's going to force its way down through this interventricular foramen into the third ventricle. So the third ventricle is filling up with fluid from the two lateral ventricles, but the third ventricle is producing its own fluid. So I'm getting lots and lots of fluid accumulating in the third ventricle, both its own and the stuff from the laterals. That fluid, as it builds up, is forcing its way down the cerebral aqueduct into the fourth ventricle. But wait, fourth ventricle is producing its own cerebrospinal fluid, so it's accumulating more and more and more here. And it's in that fourth ventricle then that the fluid is going to leave and go its separate ways. You should know that remember these ependymal cells, one of the 4 types of glial cells, line all of these cavities here. And they help, the little cilia on here, help keep the fluid moving in the right direction. Part of it is the buildup of pressure as more and more fluid is produced, and part of it is these cells helping to circulate it. There are also structures called choroid plexus. Choroid plexus are the actual tissues that produce the cerebrospinal fluid. Here's a picture. Oh, that came out pretty dark, didn't it? It's hard to see. This is a frontal picture. Thought this was going to show better. This is a frontal picture here. Here's the two lateral ventricles, and you can see these little knots, this little spongy sort of cardiovascular structure that are the choroid plexus that are sitting in the ventricles. The actual cellular structure of the choroid plexus looks like this, and it's producing the cerebrospinal fluid. To follow the general flow of the fluid, you want a picture more like this, right? And this picture in your textbook shows it very nicely. Here's some choroid plexus in the lateral ventricle. Here's the choroid plexus in the third ventricle. Here's the choroid plexus down in the fourth ventricle. And if you follow the little arrows, you can see that the cerebrospinal fluid is produced in these choroid plexus. Follow the white arrows, you can see it flowing from lateral to third, from third down the aqueduct to fourth. All of it is collecting here ultimately. And from the fourth ventricle then there are 4 exits. Some of the fluid is going to circulate right down through the spinal cord, because from this fourth ventricle there's what is known as a central canal of the spinal cord, a little hole that runs longitudinally down through the spinal cord. Some of the fluid is going to circulate right down through that. But you will also find 3 other little holes in the fourth ventricle. There are 2 lateral apertures, and this is one of them that you see right here. And then there's a median aperture that's right dead center in the back, the lower part of the fourth ventricle. And fluid in either one of these apertures can then leak from the ventricular spaces out into the space between the meninges and the brain tissue itself, that space called the subarachnoid space. And then you see little arrows showing the flow of fluid over and through those spaces. And where will the fluid go? Well, the fluid will go to where the exits are. Picture it like this room. If this air conditioning system up here is pumping air into the room, where's the air going to go as it's pumped in? Yeah, it's going to go wherever it can get out, right? If I open that door over there, where's the air going to go? It's going to go out that door, right? If I open this door over here, the air is going to go out that door. It's going to go wherever the least resistance is. Place where it can get out. And that in the central nervous system, that happens to be the dural sinuses. See, now I'm taking you back. Remember where I showed you the Dura Mater has those two layers in the cranial area. And the meningeal layer folds itself down into these little areas like this little blue part that you see up here represents what is called the superior sagittal sinus, one of the dural sinuses. It's the sinus, I put this back together here, right? If you look at the brain right here, if I put the meninges over it, you can see that little triangular space there, right, between the two hemispheres there. And so all the way along here, from front to back, there's this little triangular space called the superior sagittal sinus. And what you see is this. The blue here represents where the sinus is. You can see the brain tissue, the nerve tissue. You can see the pia mater. You can see the arachnoid mater with its web-like layer. You can see the Dura Mater is this piece right here. The fluid is in the web-like space here where the blood vessels and the webs are. And notice right here is what is called an arachnoid granulation. The dura has a hole in it, and the arachnoid mater has grown a little piece of itself out through that hole to keep it open. And fluid can literally pass right out through that opening, out through this granulation, and collect then in the dural sinus. And that's where the fluid collects. Blood also enters that from various veins. And so what I have is a collection of fluid and blood, and that's ultimately then going to connect down into the jugular veins and return to general circulation. So cerebrospinal fluid is produced from the plasma, but it comes through a special process that makes it a special fluid for the nervous system. It's produced inside the brain, in the ventricles. Pressure and the movement of the cilia move that out into the spaces around the outside of the central nervous system. And it ultimately re-enters circulation here at these dural sinuses. Anything that prevents the outflow of this fluid can lead to drastic consequences. Maybe you've heard of a problem called hydrocephalus. You ever heard of that? Sometimes, not often, but once in a while a child will be born where its head is kind of expanded. And typically it's a problem in those sinuses. The arachnoid granulations may not be formed right. And so the fluid that is produced deep within the brain cannot get out. It's blood pressure that is helping to produce it, and you can't stop blood pressure. And so the ventricles inside the brain will swell and put pressure on the nerve tissues, causing problems. Usually this results in mental retardation because of the damage to the neural tissue resulting from the buildup of fluid. And, in infants, the skull is still got a lot of cartilage to it. It's very pliable, so it can just expand. In an adult, where you've got bones that are all locked together, if you get any sort of increase in pressure or fluid, it has even more drastic consequences because the skull can't expand. The pressure just goes straight into the neural tissue and causes problems. So that's the basic layout of the cellular structure, the tissue structure, and the special